Okay. Yeah, and really finally, I really want to thank Rajiv also for the invitation. So today I want to talk about green systems biology, the need for ecological thinking. I want to discuss this with you. And what I recognized during the last hours and days here is that I come from a completely different angle of research questions. And you will see what I mean with this. And I'm really happy to talk the next days about this with you. Okay, first of all, where I'm from. So I'm in the middle of our university is in the middle of Europe, in Vienna, in Austria. It's the largest, one of the largest universities with 90,000 students. And now I'm in India. So our trip started in Mumbai, and I really have to say we experienced so much hospitality and open-minded people that this is really overwhelming. At the same time, we see all the presence of various cultures, of course, the Hinduism, so we visited lots of temples, but at the same time, you see here this white church, it's a Christian church, so it's really amazing to see really this cultural mixture and this open-minded people here. Really happy to be in India now. The department at the University of Vienna is about ecogenomics and systems biology, so I'm also quite intrigued by the fact that the center will be now called Genomics and Systems Biology. I think it's the right time for this. And I give you some examples what we understand with systems biology. The main point is we start with next generation sequencing, but our main work is really here. Proteomics, metabolomics, gene scale modeling. And in the end, we want to do some modeling in ecosystems analysis, but this is still far away, I have to admit. Main workhorses are mass spectrometers. To have all kinds of different mass spectrometers, we can actually address any kind of bioanalytical question. So I'm a chemist by education, so this is what I really love, but I can not show you any details today, unfortunately. We also recently founded the Vienna Metabolomics Center. So this is a research platform, and it's really an open research platform for scientific collaborations. So if you have any ideas about metabolomics and you want to discuss this with us, I'm totally happy to really get into contact with you, and we can start to discuss all kinds of approaches from GCMS, LCMS, structure elucidation, databases, multivariate statistics, data integration, and especially metabolic modeling also. Okay, so first I will start with the motivation of our research. So what is the motivation to actually do systems biology, then translate this into green systems biology. Then I give you examples about a proteome metabolome as a response of a genotype to the environment to measure really the metabotype, the phenotype actually, and I will actually indeed present a genotype-phenotype equation, a mathematical equation for this. And I will skip this structure elucidation for metabolomics data because of time reasons, and I will show you also applications in the field actually. So next generation sequencing, when I gave a talk, say 2015 or so, it was about 65 genome projects. I took a look today, actually it's now 170 genome projects. And it's very obvious that this is really routine methods, right? But what about the genotype-phenotype understanding? To be honest, I don't think we have a real understanding at the moment. And there was a clear comment also that annotation, gene annotation, really functional annotation of genomes is a bottleneck at the moment. So, and why do we have to put this into an environmental context or ecological context is because each genotype is somehow going through a developmental phase which produces various phenotypes which go again through an environmental phase for phenotypic selection, which then changes reproduction rates, which changes then the genotypes. So this already implements a very strong complexity in the field. So now we have also the genotypical adaptation, but also the phenotypic adaptation to the environment. And there's a nice quote While at present the purely genetical side of this problem is fairly understood, we have only to a slight extent been able to arrive at an understanding of the ecological questions involved, which means phenotypic plasticity at the natural habitat, right? And I will give you some examples of in situ data. And you guess this would be, I guess you think this is a very modern quote, but this is from 1922. So this guy, Goethe Thuresin, hopefully you know him, he defines the idea of ecotypes. If you talk about accessions, for instance, we're talking about ecotypes actually. And you see, he did not know anything about DNA structure, but he had already a quite clear idea that we have to put all this genetical analysis into the environmental research question. We do this, right? So we have all the global population structure of Arabidopsis thaliana. We recently, in a large consortium, we recently published 1,100 genomes of Arabidopsis thaliana. So it's intra-specific variation from North Sweden to South Spain. So you can imagine how strong these genotypes are adapted to environmental conditions or climate adaptation. So this is really the stressing message from my side today is, so what about the predictability of the phenotype? So here is, for instance, this is what the systems biologist is first doing once he has a complete genome. These are the 5 chromosomes here. Then you look for autologs in this genome, and then you generate metabolic networks here. I don't talk about this. It's called the stoichiometric matrix. This generates immediately a metabolic network directly from the gene annotation, genome annotation, but this does not reflect any phenotypic plasticity. It's very static metabolic network. Just to make this more clear, for instance, if you reconstruct the flavonoid pathway from grape berries and from cacao, they almost look identical, right? So there is, of course, different numbers of genes, etc., but the pathways are almost identical. So this gives you a static information, but no idea on what is going on dynamically. And this, I think I can really make very short. This is why it is so complicated, or rather unpredictability, because from the genome to the metabolome, you have a huge amplification of variables, which then finally generates a phenotype. Well, it's almost unpredictable what from a genotype to the molecular phenotype. We have even no methods to cover the full proteome, the full metabolome. Right? So there's really a lot of work to do. So I called it unpredictability of metabolism, and we have to measure the genotype-phenotype relation. Otherwise, we are not able to finally predict from a newly sequenced genome any kind of phenotypic variation or plasticity. And if you want to predict something, what do you need? It's a model. You have to design a model for prediction, and then you're already in the systems biology. And this model, this metabolic network, which I showed you, is already the starting point for each approach in systems biology. You start with a metabolic reconstruction from a genome sequence. Okay, so altogether, it's about genome sequencing, it's about modeling, and it's about the omics readout, and of course, physiology also. And this all combines in experiments and computer simulation in an iterative cycle. This is really systems biology, and this is applied from personalized medicine to bioeconomy nowadays. So what about green systems biology? Well, yeah, I want to put this into an environmental context, and you know, systems biology is very strongly represented by medical approaches and cancer research, where we have to really shift this also into the environmental sciences approaches. And why? Because this is still the primary production process on Earth. I don't have to explain this to you. It's a perfect balance between plants and us. I always call this a symbiosis on Earth somehow. But however, we introduced some side pathways which are really, really disturbing this perfect balance on Earth. And we see, of course, all these consequences, this climate change, and of course, plant productivity and quality, etc., etc. So this then finally led to the idea of the megatrends, the food, feed, fuel, and fibers. Every day in the newspaper you can read some information about this. Department of Energy is really concerned about energy genomics, sequencing everything from plants to microbes with respect to renewable energy. India invests a lot of money into agricultural fundamental aspects also. And at the same time, we have natural genetic variation in Arabidopsis thaliana. But now comes our connection here also to ICRISAT. Of course, we are more interested than really in applied natural genetic variation. And here we collaborate with Rajiv on the Pearl millet genome and also on the physiological and molecular data. So this is what we are doing at the moment, a molecular physiological model of a Droughtstressed perception Pearl millet together with Rajiv. And here I will present in the end some ideas on this. Okay. So this constitutes the whole framework of green systems biology. This was already also mentioned here at the Institute. We really face a new green revolution, and we can expect a fundamental genotype, environment, phenotype equation now. And this has to be put into a socioeconomic context and in a biological context, from ecology to evolution. And we have also sessions here, for instance, in the Metabolomics conference, we have sessions on this topic nowadays. Okay, so back to my main point. So genotyping is great, but it gives you no causality, no physiology. So this is a quote from Craig Venter. Will soon become a commodity, and we are already there, I think. But then genetic variation with physiology is a challenge as great as genomicists faced a decade ago. I think it's an even more stronger challenge. That's why it is so important and timely also to think about these systems biology approaches. Now, what we are doing actually is to fill the gap here. Is we developed a large platform with proteomics and metabolomics approaches here. And now I give you lots of examples. So we really do environmental studies, but also, of course, controlled experiments. I give you some examples of tropical plants also. So this was an approach with potato breeders together. So here, at that time, it was not clear that we could translate our proteomics and metabolomics method really into the field. Is this really stable enough? Are the technologies stable enough to identify robust biomarkers, for instance? So we worked with potato breeders together. They had lots of cultivars with very defined properties. And then we had to develop a new approach. I skipped this because this is technical details. I'm totally happy to explain this to you. This was the only way that time to start to assemble 500 or 1,000 shotgun proteomics data, and we published this in Nature Protocols as well. So first of all, it was really possible with a very low number of misclassifications to separate all the cultivars in field conditions, so in different plots grown in Germany. And with various statistical methods, we gave then the breeders very strong protein markers related to black spot susceptibility or so, or to chip quality. So this was really possible that time. And we based on this method development, we apply also this method also in metabolomics now. So another interesting analysis was then development. Actually, now we really are interested in developmental processes. This is work by Palak here. She was working on pollen proteomics, and as you know, the pollen is most sensitive. The male gametophyte is the most sensitive part actually for productivity. So she was interested in the proteome during development. She isolated various pollen stages here from microspore pollen mother cells to mature pollen, did proteome, so we had a very strong change in the proteome. But what was really intriguing was that she has seen already in the very early stages in the pollen mother cells a very high proportion of stress-related proteins without any stress treatment actually. So they are already perfectly prepared for sudden stresses, and we call this process then developmental priming. And Palak will also present these data on a poster. We also went for cacao seed development. Now, I come back to my example, grape berries cacao seed development. We applied the large proteomics metabolomics platform here. This is a typical, only a selection of metabolites which we see here. This is a primary metabolism, sugar, secondary metabolism. We have seen a very sophisticated balancing between these secondary metabolites and phenylpropanyl amino acids compared to the flavonoid pathway, anthocyanin pathway. And if you now combine this with the proteome data, you see really these simple phenols and phenylpropanoids start very early in the development, and then flavonoids biosynthesis start later on. We did similar analysis with grapeberry development. We were interested in the flavonoid pathway. We see here a very clear distinction in the flavonoid pathways of flavonols and anthocyanins. For instance, we did the same on proteome data. And now the main point is that now we start to fill these static networks here with a very strong dynamic process. So you can really distinguish each developmental stage very, very clearly and then look also for the dynamics of this system. So this is how we really start to fill the gaps, the genotype-phenotype gap. We did this also with lotus seed, and all the data are stored in a public available database. Okay, so one example about metabolomics I want to give today, because this is also an environmental approach. So we're really doing here field work. So metabolomics, I don't have to explain it, large platform here with various mass spectrometers, and we collaborated with people from biodiversity research in a Vienna experiment in Germany. So each plot here is a classical grassland community, but sometimes are monocultures. So this is only one species up to 60 species. And this is, of course, a perfect setting to see how the metabolome can actually predict biodiversity, or how biodiversity actually impacts the metabolome of a plant. So we selected some species, we applied a large metabolomics platform. And first of all, you see, if you now, this is a principal component analysis, you see you can distinguish each species very well based on the metabolome. This is not trivial result, because if you take textbook knowledge, or if you take a metabolic reconstruction from the genome, you would not be able to predict the distinct dynamics in the metabolome of these different species. That's a very, very clear point here. Now we were interested in the diversity effects on the metabolome, and here we see, if we just look for one species, we see a clear grouping, so two groups here, and this species is highly influenced by the presence of legumes in these different plots here. So they are clearly distinguished, but this is not only a classification process, but we also give the biomarkers for this. So here it's a clear CN balance biomarker signature, right? So you have here increasing amino acids, you have decreasing central metabolites like alpha-ketoglutarate or maltose or raffinose, etc. So you have immediately a metabolic signature. We call this the legume effect, or the legume signature here, and they are highly distinguished based on the presence of legumes in the The biodiversity effect was most pronounced in the secondary metabolism. So here you see a clear gradient in secondary metabolites in dependency on the species richness up to 60 species. And then we gave some assumptions. So what are these secondary metabolites which really are responding to the biodiversity effect in these fields? Most important, we have seen a metabolic signature for each individual species. So there's not a general metabolic signature for all the species, but really each individual species has a very specific signature. And this is something, it's contradictory to classical biodiversity experiments, and I think this gives gives a new layer of information now on the metabolome, on the physiolome of a plant in the natural environment. Okay, have to skip this because of time reasons. So we did a lot on structural elucidation of these metabolite data. This is really a bottleneck nowadays in metabolomics. So we have thousands of so-called features which cannot be identified, and we did a lot of structural elucidation. However, we can use all these features also for classification. So here, for instance, we had a collaboration with Spanish people on pine. Pine is a very important commercial species in Spain. So you see here all the different genotypes or ecotypes, if you want. And the question was, can we identify the region of origin of a pine from its metabolites here, distributed over Spain and France and North Africa? And yes, it is possible. So we measured the metabolome and we distinguished really various different ecotypes here, and we classified really parameters like growth or rainfall or aridity very strongly with the metabolome also. So my other part of the approach is the modeling approach. So here I've shown you now many examples about the data. So this is pure data. So you generate protein data, metabolite data, transcript data, physiological data. This comes into a data matrix here, which gives you directly rise to very sophisticated statistics. So you can do a classification, but you can do also physiological or functional interpretation of the data. Now the question is this in any way related to the genome sequence here? Everything is based on, in principle, dynamics of the system. This is a time-dependent change of a metabolite here, or a protein, or a transcript, or photosynthetic rate, and they are all coupled to each other. The problem is we don't have any idea about the system's equations in this system here. This is not known. But over the last 10 years, we developed an equation which actually links this system's equations with the data. So here, on the one hand, we have the covariance data. What I showed you, we can do immediately statistics with this covariance data. And the main point is the Jacobian here, J, and this Jacobian is actually identified or defined by the stoichiometric matrix from a metabolic reconstruction. So, and here we have a fluctuation matrix, it's D, and this is an environmental fluctuation, which we see actually in the environment. So this equation really links the genome sequence, especially the metabolic reconstruction, and the data-derived dynamic model of the phenotype. So I put this here in this context now. Here, J, Jacobian, is derived from the genotype, from the metabolic reconstruction. The covariance matrix is what we measure actually to define the dynamic phenotype here, and the D is the environmental perturbation, and then we feed this back. Now, nice equation is very academic thing. The main point is, can we really use this also? And indeed, we can use this equation. We implemented this in a toolbox. We call this covariance inverse. So we really start with the covariance data and go back to the phenotype. And this is also a toolbox for any kind of data mining. So I'm happy to explain this more to you. This is a graphic user interface, so you can upload any kind of data, transcripts, proteins, metabolites. You can combine the data. You do statistical analysis. You do normalization, transformation, etc., etc. We implemented this equation here, and we can indeed calculate back metabolic phenotypes. So here we used Arabidopsis thaliana under 16 days of cold and light stress here. And now we measured the whole metabolome here. And what you see, if you just unsupervised plot this in the principal component analysis, you see immediately the trajectory of the stress over the metabolome here. So now the question is, can we derive any metabolic phenotype by this? We applied the equation here. And now by solving this equation here from the metabolomics data, we see in a heat map metabolic perturbation points, and they point directly to biochemical pathways. So we have really clear ideas now of what happens. So for instance, the starting point of flavonoid anthocyanin biosynthesis is highly perturbed in the system from here to here. It's obvious, right? So you have here high accumulation of anthocyanins, but at the same time you see photorespiration is strongly interacting with the flavonoid pathway, and we have proteins, so polyamines are also strongly perturbed in the system. So now we have really a system where we can understand the metabolic phenotype directly from the metabolomics data. This is another system where we put the plant under energy starvation, extended night here. We solved the system. We predicted a perturbation in the pyruvate metabolism. We measured pyruvate metabolism, and indeed we found under energy starvation the pyruvate dehydrogenase is downregulated, and this makes totally sense. If you have a plant under good conditions, you have photosynthesis, but if you put them under low energy, you deplete you increase the respiration, you deplete the carbohydrates, and then there must be a regulatory point which then helps the plants, and this is exactly the pyruvate dehydrogenase. So this is really possible now to predict the biochemical phenotype. So there is a clear link between the genotype and the phenotype here with the equation. I'm coming to an end now. And we apply this also in situ. So we have, we have in situ samples from Arabidopsis thaliana. We do metabolite profiling. We see they are completely different. So this is now intraspecific variation. If you now plot a correlation network, these metabolomes correlate with GPS and climate parameters, but you have no clue what is a biochemical phenotype actually. And if we now apply this, we really see, for instance, we have two different natural populations, and this is in situ data, in-field data. We clearly distinguish these different natural populations, same species, Arabidopsis thaliana, based on biochemical pathways and perturbations here. We integrate this with enzymatic activity, RNA-seq proteomics, and also signaling analysis, phosphoproteomics. And just one minute left. We also apply this now to Penicetum glaucum together with ICRISAT, and here we design the first molecular physiological model of drought-stress resistance. So this was recently published in 2016 by Arindam Ghatak, and here we applied this drought system with sensors here about soil water content. We see stress. Water stress immediately reacts on the stomata conductance, so you see clearly stomata signals here. We see at the same time a very stimulated root growth, and if we now measure all the proteomes of these different stages here, and we have tissue-specific proteomes, then we can generate a model here, a molecular physiological model of drought stress perception, that we have an increased root growth, and we have lots of perturbations in signaling pathways, which gives signals between the shoot and the root in the system under drought stress. Okay, and we have proteins correlated to the harvest index. I think this is something we want to work further on. And applying all this to the natural genetic variation will be really the bottleneck in the future. So in summary, I think we will reveal a complete new picture of the genotype environment phenotype equation. We can even calculate the metabolic phenotype from our data. And we start really to understand the phenotypic plasticity in situ, so really in the natural field. And this has to be applied to crop plants and natural ecosystems. And yeah, I'm very much looking forward. I think it's really an exciting time for all of us. I have to thank, of course, my group, and especially Palak and Arindam for this great hospitality and guiding us through India. Thank you very much.
A little bit over time, so maybe if there's one burning question we can take. Otherwise,
I'm curious to ask one question. You showed one slide that metabolome very specific to one different color of one species, flowering plant.
Yeah.
If it is a variegated flowering plants, would that also work for it?
Excuse me.
If it is a variegated plant with the different color variations,
yes,
would that still metabolome would be specific, or what would you expect?
Yeah, you would have metabolic signatures very specific. So classification is very strong.
Okay, thank you. Mm-hmm. Thank you. Thank you very much for an exciting, high quality, knowledge rich presentations from the eminent speakers, and they kept all in time, as Rajiv instruction is always there. And thank you very much once again.